All right, hello there, and welcome to Cell Biology, where we're going to combine lessons 8 and 9, just because they're both on the same topic, and lesson 8 includes what would have been a practical. And obviously, since we can't do that, we're just going to move on. So turn to lesson 8 first. We're going to go by it in probably about 5 minutes, and then the rest of it will be on lesson 9. Okay. With lesson 8, I do still want you to do the do now because it does kind of remind you of things that we've learned about in lesson 7. So go ahead and pause the video and get started on those. Perfect. Let's start off with number 1. So number 1 asks, define diffusion. So all of last lesson we were learning about diffusion, what it was, some of the factors. So let's go through this, all right? Diffusion is the movement of molecules from a high concentration to a low concentration. Okay, that's kind of the big broad definition of diffusion, and it will become important as we talk about today's lesson. Number 2, state 2 factors that affect diffusion. And number 3 actually asks pretty much the same thing, just 2 more factors, right? So last time we learned about 4 specific things that determine or change how fast diffusion actually occurs. So we call this the rate of diffusion, all right? How fast or how slow something can be. So the 4 that are mentioned in last video, it would be temperature and concentration gradient. Remember that concentration gradient just means the difference in concentration. So bigger concentration gradient means you have a very, very big difference between the high and the low concentrations, which means that the rate of diffusion will be faster. With temperature, remember that the more or the hotter something is, the more energy it actually has. So more energy means more movement. The other 2 will be surface area and thickness of the membrane. So the analogy that I gave you last time for the surface area was you're in a room with 4 walls and you have 4 doors on, you have a door on each wall. And increasing the surface area is kind of like adding more doors to those walls, okay, so that there's more space or more entry points into that room. With the thickness of the membrane, the analogy that I use is a really, really thick shirt compared to a really thin shirt, right? So thick shirts, it takes a while for it to get wet, and then thin shirts, it's really easy to tell if it gets wet or not, okay? All right, number 4, suggest which states can move by diffusion. So there's 3 states that we talked about, but only 2 of them can actually go through diffusion, and that would be liquids and gases. Number 5, suggest what is meant by concentration gradient. So I just kind of mentioned this when we were talking about number 2, and that is the difference in concentration between 2 sides or 2 points, okay? Number 6, in terms of diffusion, suggest what passive means. So we know that diffusion is called a passive process. And what that means is passive means that there's no energy involved. Okay, so you don't actually need to stir the glass of water with food coloring in it. It'll just happen by its own. But obviously adding in energy into that system, okay, so actually stirring it, you are putting in mechanical energy, right? And so that energy is then actually allowing the food coloring to get diffused even faster. All right, but the important thing here is that diffusion itself is a passive process, so there's actually no energy that's actually required. Number 7. Oh, sorry. State the 2 gases that are exchanged in the alveoli. Remember that alveoli is part of your lungs. It's the air sacs, and we're going to talk a lot about that today. And the 2 gases that we talked about are oxygen and carbon dioxide, which are 2 gases that are very easily associated with the lungs, right? You breathe in oxygen and you breathe out carbon dioxide. Number 8, suggest one adaptation of alveoli that enables efficient gas exchange. So this is kind of going to be like an evolutionary thing, right? So what is it about our alveoli that allows us to do this gas exchange really efficiently? And that is, we have a large surface area. Okay, and I'll show you pictures of this today. Right, so just make sure that you have all the correct answers before we move on. Perfect. So with lesson 8, we are going to talk about osmosis, all right? So I'm not going to give the definition of osmosis yet, but the way that this was supposed to work was that you were supposed to do this practical on osmosis, and then we would have had a lesson on it, Okay, so in lesson 8, hopefully you would have known to define the term osmosis and give examples and investigate the effect of range of concentrations of salt or sugar solutions on the mass of plant tissue. So take a look at the YouTube video link that's on there. This is the practical that we would have done had we been in school. And basically what it is you get a potato and you cut it up, and you would basically immerse the potato in different solutions of sugar, so different concentrations of sugar, and you would see which potato has the most water content inside it. All right, and how you would do this is you basically weigh it. So the heavier it is, the more content that it absorbed, okay, or that would diffuse into the potato. Okay, so that brings us to lesson 9. All right, so like I said, watch the video on YouTube of the practical before you start this. All right, and that way you have a little bit of a better idea of where we're coming from for osmosis. All right, but that being said, on lesson 9, turn to the do now and get started on those. And this one, I'm not going to give you as much time just because there are some things in here that we have not mentioned before in previous lessons, but hopefully you remember from previous years. So this might be a little bit more of a review than anything else. Okay. Right. So the first 3 questions have to do with variables. So you have your independent, dependent, and control variables. These are actually really important to understand because on your GCSEs you will have questions that give you an experiment and ask you what's the independent variable or what's the dependent variable, and you need to identify which one is which. So let's make sure that we know what each one is. So define independent variable. The independent variable, it's kind of self-explanatory with the word independent. If you are independent, then you don't rely on anyone else, right? You make your own money, you don't ask for help, you're very independent, right? So that variable is the one that you control, or it does not depend on another factor, all right? So this one will be something that you can change throughout the experiment, and no matter what you do, it'll stay fairly constant. So the experiment that I like to use as an example is growing a plant, right? If you had an experiment where your question was, what are the best conditions, or maybe how much water does a plant need in order to grow really well, okay? So your independent variable in that one would be the water that you give. So you might give one plant 10 millimeters of water. You might give another plant 20, then 30, then 40, then 50 mils of water. And you would see which one of those plants grows the best by measuring maybe how tall it grows. Okay, so water is your independent variable because it doesn't depend on another variable, right? That would be your dependent variable, which brings me to number 2. So your dependent variable is the one that is being tested. So in the same experiment where you're trying to figure out, well, what amount of water is best for a plant to grow? Your dependent variable is the one that responds to the independent variable. So if you're changing the amount of water that you're giving to a plant, okay, then your dependent variable will be the growth of that plant. Okay, in this term, or in this example, we want to make sure that we have numbers to show our results, right? So that's why I said we would measure how tall a plant would grow, and that would be your dependent variable. That changes according to how much water you give it. It's dependent on the water. Okay. So then number 3, what is this control variable, right? Because I said that your independent variable is one that you control. So is this different? And the answer is yes, they are. Your control variable is something that is kept the same throughout the experiment. Okay. So what I would do then in this experiment, I might change a little bit and have 30 mils as my control. All right. So if there is a known fact that 30 mils is the best amount of water, and you just wanted to see, well, maybe 40 mils is better or 50 mils is better, then 30 mils would be your control variable, okay? Or you might choose your control variable to be what happens if you don't give water to any plant, right? So then you would have another plant that you would give no water to, and that is, if it doesn't have water, it's going to die, okay? That's your control. You know what the result is going to be, okay? All right, so number 4, again, this is basically what we would have done in the practical. And so osmosis goes from a dilute solution to a more concentrated solution. I'm gonna teach you guys what this actually means when we get into the lesson. Number 5, state what the term valid means. So going back to experiments, if something is valid, if a result is valid, all right, that means it has credibility. All right. If I came up to you one day and I said humans can fly, right? You might first say, no, they can't because I can't. But if I told you that I have proof that humans fly, you would want to see that proof, right? But you already know that it's not valid because my results are probably going to be very skewed. It might be false. I might have tested it wrong. So valid just means that it's acceptable as a result, or it has credibility. All right, so your experiment or whatever you're trying to solve or figure out, it has to be valid. It has to be credible, right? Number 6, state what a control is used for. So going back to number 3, a control variable is just to make sure that your experiment is valid, right? So going back to the watering the plant one, getting rid of the water or having a plant as a control where it receives no water, that makes sure that, okay, well, we know that water is essential for the plant to grow, okay? Number 7, state how we know an experiment is reproducible. So when you look at the word reproduce, that means to kind of multiply or to create children, right? So when an experiment is reproducible, that means we can reproduce, right? We can produce again the same experiment, all right? So this experiment that I'm talking to you about with this watering plant one, it is very reproducible because anyone can go buy a couple seeds, right, and get water and do this exact experiment. But there are certain experiments that are not reproducible in certain areas, right? So if we talk about space, right, a lot of the experiments in space are not reproducible because maybe they rely on a specific event, okay? And that way, the more times that you do an experiment, the more valid it becomes, right? Because if you prove it over and over and over again, then you start to think, well, maybe it's going to happen every single time. Whereas if you do an experiment once and take it as truth, right, then you don't really have the credibility to say that that thing is actually true, okay? So just keep that in your minds that the more times you do an experiment, the more credible it becomes, right? And you look at the results, and whatever happens the most, you can kind of bet that that's what's going to happen, 80% of the time, 90% of the time, okay? Number 8, state what accurate means, right? So if something is accurate, we're talking about the results, all right? And also about how you record the data. So with the watering plant one, if you want to be accurate, then you really want to be accurate with the measurements that you do with measuring out the water, measuring the height of the plant, all these different things. You need to be accurate so that your results become more valid. Okay. So sorry that was a little bit longer because I have to explain a lot of these things, but it will help you in the future. All right. Okay, so now we get to what we talk about osmosis, right? We need to learn about the term osmosis, give examples of where it occurs, and also we're going to learn about how the range of concentration, right, will have a difference on the mass of plant tissue. Okay, so we're going to talk about this as we go forward. All right, so let's get into osmosis. So let's start off in the first line here. It says, last lesson we looked at diffusion. Osmosis is a specific type of diffusion just for water molecules. So that gives you kind of the initial idea of what osmosis is. It's really just diffusion. That's all it is. Osmosis is a name that we give to the diffusion of water molecules. That's it. And you might think, well, that seems kind of weird. Why can't we just call it diffusion? And there is a reason why, because water behaves very differently from a lot of different molecules. Okay, and also water is kind of the universal solvent in our lives today. So just take it for what it is. So here's the first definition of osmosis. Right. So osmosis is the movement of water from a dilute solution to a more concentrated solution through a partially permeable membrane. All right, so I want you to highlight that right there because this is important. if something is dilute, all right, there's a picture on the bottom here. If something is dilute, then it means that there's a lot of water compared to the amount of solute that's in it. All right. So imagine a cup of water and you put some salt in it, right? If you have a large glass of water, all right, and you put 1 tablespoon of salt, you're going to have a pretty dilute solution. And what that means is you can still taste the salt, but it's not very strong. Compared to if you had the same glass of water and you put in 10 or 15 tablespoons of salt, then you're going to have a very concentrated solution. All right. So if it's concentrated, then you can really taste that salt. All right. And it doesn't have to be salt. It can be like juice, right? You can have very dilute juice. If you're using squash, right, and you don't put enough in, then you might taste more water than the actual squash. As opposed to very concentrated. If you just drink the squash with no water, right, it's going to be extremely strong. Okay. So, where does osmosis occur? Well, osmosis occurs in all cells to enable a constant water level that is to be achieved, right? So I want to focus on this term constant, right? If something is constant, that means it's the same everywhere, right? So you can say that breathing air is a constant, right? Because everyone has to do it. In this term, where it says osmosis occurs in all cells to enable a constant water level, right? If you want a constant water level, that means everywhere in the cell has to have the same water content, okay? So if 1/2 of the cell has 50% water content and the other side has 90% water content, right? Then you have to change the amount of water that's on both sides so that everywhere is the same. And there's a term for this, a term that you've heard before, and that term is equilibrium. So when you have a constant level throughout the whole system, all right, and the system we're talking about is the cell right now, this is called equilibrium. All right, so the concentration of water has to be the same throughout the whole cell, all right, or else it might be more difficult for that cell to survive. Right? And the same thing happens in your body as well. If one part of your body has too much concentration of anything, right, you might actually feel it. Whereas, if you look at your individual cells, the same thing has to occur. All right. So I'm just going to explain this dilute concentrate a little bit more because I want to make sure that you understand this before we move on. So dilute means not a lot of solute. Okay, and solute is whatever you add into the water. So in the example that I gave you, I used salt, but you can use squash, whatever you want to use, okay? But when something is concentrated, you have lots of solute compared to the water, all right? Okay, so in the cell, I'm starting on line 9 again. Water molecules will pass both ways through a membrane, and the action is passive and needs no energy, all right? So we're still talking about diffusion, right? Osmosis is a form of diffusion. So you still need no energy, right? It's still passive, and water molecules can pass through both ways. So if a cell contains little water, then the solution inside it will be concentrated and osmosis will occur into the cell, right? Because if you have not a lot of water, but a lot of solute, right, it's very concentrated, right? So think lots of salt, not a lot of water. So then you need to add water to it so that it becomes more diluted. And then vice versa, the opposite is also true. So if a cell contains lots of water, right, it'll be very dilute inside. So then water has to leave in order for it to become that middle area there. Okay? So let me show you a diagram. So this one, you are presented with a beaker and there is a permeable membrane in the middle, right? So remember that membrane is kind of like a skin, right? Certain molecules can get through, but certain ones can't. So in this scenario here, you have basically a sugar solution, all right? Water and sugar. And you'll notice on solution 1, the left side, there is a lot more water molecules than sugar molecules. Okay, so the sugar molecules are the big orange ones, okay, or big red ones. On the right side, solution 2, you have a high concentration of sugar, right? So if you compare them, you have a lot more red molecules than the water molecules. All right, the legend's at the bottom there. So what's going to happen? That means that the molecules aren't going to move themselves, right? The sugar molecules aren't going to move because they're too big to go through the membrane. So the water molecules will then go towards the right side. Okay, so then what you have is a diffusion of water going from the left to the right. Now you might think this is weird because the water level is pretty, it's at the same level, right? And the interesting thing is this will actually happen. Water will actually move so that the level becomes unequal, okay, in terms of height. But the concentration will become the same throughout. So to illustrate this a bit more, this is an exaggerated diagram. So it's not going to be this extreme, but I want to show you this to kind of show you this is essentially what happens, all right? So same thing. On the left side, you have a very weak solution or a dilute solution. On the right side, you have a very concentrated solution. And when it goes to the right side of the diagram, you'll notice that the concentration is the same on the left and the right side, even though the amount of solution that's there is different, right? So this, I think, does a good job at explaining that concentrations are very different, all right, in the beginning. But what osmosis will do is the water molecules will then move to the area, right, that is more concentrated to make sure that the concentration levels are at equilibrium. All right. So now we get to talk a little bit about, why is osmosis so important in your body? Okay. So osmosis and diffusion will be affected by the surface area of the cell/organism involved, right? So again, there are things that will speed up osmosis and diffusion, right? One of those being surface area. And we have a little bit of a learning activity later on to show you what I mean by this. The next sentence here is actually quite important. The larger the organism, the smaller the surface area to volume ratio. So if you compare yourself to a single cell organism like bacteria, just one cell of a bacteria, you are humongous compared to that cell, right? But what's different between you and the cell is that cell actually has a very, very big surface area to volume ratio. And what I mean by that is that bacteria cell, its entire body can diffuse water or molecules in and out of its cell. Okay? Whereas yourself, you're made up of billions of cells, multicellular cells, right? Every cell in your body is different. And what that is you can't diffuse things out of your skin, right? It needs to go through a channel or a system in order to actually diffuse into your body. So the amount, considering how big you are, right, your ratio of surface area that can diffuse to the volume of yourself is actually quite small, okay? So this kind of presents an issue because we need a lot of oxygen, we need a lot of energy, right, in order to actually do things that we need to do. So in order to bypass this, evolution has given us a way for cellular specialization, right? So in different parts of our body, we have different adaptations or evolutionary things that help us absorb as much as we can. All right, and the two places we're going to look at today are your intestines and your lungs. All right, so two structures in each one. In your digestive system, you have these things called villi, and this will increase the surface area, as well as your lungs. Your lungs include these things called alveoli that are folded in structure to increase the surface area. So your alveoli is actually located right here in your lungs, all right? And they look like these little sacs, okay? And inside the sacs, you'll notice, right, or not inside, but around the sacs, you'll notice that there's two lines of red and blue. Those represent your arteries and your veins, okay? So when you breathe in, the oxygen molecules would diffuse into your bloodstream from your alveoli, okay? And your alveoli is able to do this because it has such a very, very big surface area, okay? So this next sentence here is actually really important. So I wanted you to highlight it, put a star next to it, make it stand out. By increasing the amount of surface area, you increase the area available for diffusion to occur over, and therefore increase the rate of diffusion. All right? So, if this isn't hitting home yet, then hopefully, this sentence will make it go there, because the thing I want you to understand is that the bigger the surface area, the faster the rate of diffusion. All right? So go back to that analogy of the room with 4 walls, and you have a door on each wall. You have the entire school wanting to get into that room, but there's only 4 doors, right? And so, students getting into that room is going to be very slow. But if I added more doors, right, more entryways to get into that room, then the rate at which students enter the room will increase, right? It's going to get faster. All right? So that's your alveoli. I'm going to show you a picture of your intestines. So if I took a microscope and I went into your digestive system, especially in your intestines, this is what I'm going to see. Right? So it kind of looks like a cleaning mop almost, right? And there's a reason for this. Because in every one of those little structures, right, the little tube structures poking out, there are, same thing, arteries and veins that go into those structures where then it can absorb the food molecules that are in your intestines, okay, as well as water. So then you have millions of these inside your intestines, okay? So if you compare this to just one hollow tube that doesn't have any villi, then you're actually not going to absorb as much as you think you would, okay? So these actually allow most of the food and water to be absorbed into your body. Okay. All right, so now we get to this portion of the video, of the lesson, where it says, if you consider the experiment from last lesson, right? If you cut the potato up into smaller pieces, do you think the rate of osmosis will increase or decrease, and why? So honestly, you actually don't even need to know what you did for the experiment, because it's asking you, if you cut up a potato into smaller pieces, are you going to increase the rate of diffusion or decrease it, and why? And the thing is, it's asking, what are you doing to the surface area? All right. So again, you might not have the same answer as me, but go ahead and try this question really quickly. All right. Write down what you think is the correct answer, and I'll show you what I wrote. All right, so even if you don't have the exact same words I do, don't worry about it, as long as you have the gist of what I'm trying to write. So, the rate of osmosis will increase because cutting the potato up into smaller pieces will increase the surface area. When the surface area is increased, then there's more space available for the molecules to diffuse. All right, very simple. Now, you don't need to go into detail about this, but that's essentially what it is. And I'm going to show you through maths that cutting up something will increase the surface area, okay? All right, what we have here is basically a table that shows you 1, 2, 3, 4, 5 different cubes, okay? And I'm going to show you that we're going to keep the volume the same. All right, we're going to keep the volume the same, but I'm going to show you that by getting smaller and smaller cubes, we're going to actually increase the surface area. All right, so let's read off the top part here. Surface area is an important factor in many chemical processes, some of them natural, some man-made. Either way, increasing the surface area is a reactant, of a reactant will often increase the rate of reaction. And you're going to actually go more in depth when we talk about chemistry. But the best way I can explain this is basically, if you have a large cube of sugar and you put it into a hot cup of tea, right? You stir it around, it takes maybe a minute for it to dissolve, right? Compare that to putting powdered sugar in, right? Or granulated sugar. That one will dissolve a lot faster than the cube of sugar, right? And that's because what you did by putting it in powder form is you've increased the surface area. There's more area of it reacting with the hot tea in order for it to dissolve, okay? And with diffusion, it's exactly the same thing. So let's go through the first row together. All right, so you have one large cube, and if you know anything about a cube, it's a square, right? All sides are a square. And finding the area of a square, right, you just basically multiply the two lengths together. So one length of the cube is 16, right? It doesn't matter if it's 16 centimeters or 16 meters, we're all doing the same thing, okay? So the length is 16. So then the area of one side of that cube should be 256, because all you have to do is 16 times 16. Now, in column C, it asks you, what's the surface area of the cube? And to get the surface area, a cube has 6 sides, right? 1, 2, 3, 4, 5, 6 sides. And each one of those sides has an area of 256. So what you're looking at is basically one side has 256, another side has 256, another 256. So you're adding 256 6 times. So you basically multiply it by 6. Okay? And 256 times 6 is 1,536. Okay? So that's the surface area of that one cube right there. Now column D asks you, what is the volume of a cube? So volume of a cube or a 3D shape like this is actually quite simple. You multiply the base times the width times the height. All right. Some people use length times width times height, but both work. So in this one, all sides are the same, right? So basically you would do 16 times 16 times 16, and that would give you 4,096. Okay. So you have one of these cubes, right? We're looking at column E. We only have one of these cubes. So what is the total surface area? It's the same as C, right? 1,536. And G asks, what is the total volume? All right, so this one, same thing. You only have one of them, so it should be the same. And then it asks you, what is the surface area to volume ratio? And what you do is you put 1,536 by 4,096, and it looks like this. Okay, it's a ratio. So that's the first row right there. The second row is going to be a little bit different. Okay, so now we have a smaller cube, a cube that is half the size of the first one. So instead of 16, now we have a cube that has 8 by 8 by 8. So then the area changes as well. Instead of 16, we're now multiplying 8 by 8, which is 64. And so if that's the case, then our surface area also gets smaller. But you might think, well, Mr. Chung, you told me that cutting it up is going to increase the surface area. Just wait, all right? We're just looking at one cube now. So the surface area of one of these cubes is the same thing. You multiply the area by 6, all right? So 6 times 64 is going to be 384, which is a lot smaller, right? But just wait. Now the volume of this one cube, right, is going to be a lot smaller as well. So volume, same thing. Length times width times height. 8 times 8 times 8. And that is 512. Okay. Now you're probably thinking, well, this doesn't make any sense. You told me that cutting it up will increase the surface area. Now remember when I told you in the beginning that I need to make sure that the volume of this is the same. That's the only way to make it fair, right? If I cut down the cube and I'm only counting that one cube, then obviously, yes, right? The volumes and the surfaces are going to be smaller. But look at column E, okay? We have 8 of these cubes, all right? So what you need to do then for column F is you have one cube. One of these cubes has a surface area of 384, but you have 8 of them. So now you multiply 384 by 8, and that gives you 3,080. Okay? Almost double what you had in the first one. So then what is the total volume? Same thing. The volume of one cube is 512, but you have 8 of them. So then your total volume is going to be 512 times 8, which is 4,096, which is the same as the first one, right? So essentially, column G should all be the same, right? Your volume should always stay the same, okay? And what you're going to do now is you're going to actually, you're going to multiply, right? Or sorry, you're going to make it into a ratio, right? Your surface area to volume. And that's going to be 3,080 to 4,096. Okay? Right. So for the rest of them, I want you to try them by themselves, all right? So the last 3 rows, right, you're going to do by yourselves, and you're going to let me know how it comes out, okay? Use the hints at the very, very bottom there. So the ones in the red box, okay, it tells you basically how to do each column. So for column B, you want to multiply the 2 lengths together. Column C, you want to multiply the cube, or sorry, you're going to multiply the area by 6, okay, and all the way through to until column H. All right, so what I'm going to do is I'm going to show them row by row, and you'll notice that the volume will stay the same, but the surface area will actually get bigger and bigger and bigger. Okay, so what we've done in the next one is we've reduced it to 4, all right, but we still want to keep the volume the same, and so this is what your row should look like. The area of one side of the cube is 16, the total surface area of one cube is 96, but you have 64 of those cubes, so then you get a total surface area of 6,144. Okay, and your volume as well, it's a lot smaller for one cube, but when you have 64 of them, then you still end up with 4,096. Okay, and we get even smaller. All right, so there's your next one. The total surface area of a cube that small is 12,288. And the last one, if you have cubes of 1, 1 by 1 by 1, you're going to get a total surface area of 24,576. All right. Now, the really, really important thing to notice here is that your volume, right, your total volume does not change. All right, because we still want to work with the same amount of whatever we're putting into our solvent. Okay, but your surface area, as you cut it up into smaller and smaller pieces, the surface area actually increases by double almost every time. Okay, so hopefully this helps you understand that increasing the surface area, right, or cutting up a thing smaller and smaller and smaller will increase the surface area. And when you increase the surface area, you are increasing the rate of diffusion or the rate of whatever you're doing with that chemical. Okay, so there we go. And that brings us to the last portion of our video where we do the knowledge check. All right, so as always, do the first 8 questions, and the last 2, if you feel brave enough, try them out, but I do go over them together, all right, because they're a little bit more of a thinking question. Okay, so pause the video and go ahead and do that. Perfect. So number 1, going back, what is osmosis? Okay, osmosis is the movement of water from a dilute solution to a more concentrated solution through a permeable membrane. So the 3 words that you absolutely need to know there in order to make sense of this is dilute, concentrated, and permeable. Okay, so make sure you know those 3. Number 2, define the term rate. All right, so rate will be the speed at which something occurs. All right. So if you have a very fast rate, then obviously it goes fast. Low rate goes slow. Number 3, identify what a partially permeable membrane is. So a membrane is the organelle that coats the entire cell, controls what goes in and out. But if something is permeable, that means something can go in and out, right? So something that's partially permeable means that only certain molecules are able to pass through. And notably, this is going to be probably the ones that don't require a specific size. All right. Number 4, state what variable is drawn on the x-axis. So with these questions, I would have had, in the experiment, you would have drawn a graph showing me the rate of diffusion, essentially. So on the x-axis, you actually draw the independent variable. Okay, an independent variable can be something like time. It can be the amount of water. It can be temperature, right? These are all things that generally you would control in an experiment to see how it affects your dependent variable, right? And then on your y-axis, that's the one that's going to show you all that change, okay? Number 5, state 4 things a graph needs. Now, this could be quite different, but from my expectation, I would expect every student to have these 4 things on a graph if they were to submit it for work or on an assessment. And that is, you need a title. Okay, what am I looking at? The y-axis label. Okay, so remember that y-axis is the one that goes straight up, right? And the way that I remember is y points to the sky. So your y-axis label has to be something, like height, right? Height changes with the amount of water that you give a plant. You also need an x-axis label. So what is your independent variable, right? These other 2 things are ones that depend on the experiment, but I would say that your scale is quite important. And your scale is basically how what your increments are on your x and your y axis. So if you're measuring the height of a plant, right, depending on how much water you give it, then your scale might be in centimeters, all right? Because you're not really growing a tree, right? You're growing a plant. And you wouldn't really use kilometers when you're measuring the height of a plant. With the legend, right? You need to, if your graph has multiple bars or multiple lines that show different plants or whatnot, then you need to tell me which one is which. Okay, so the bottom line here is that your graph needs to be as have as much explanation as possible. Okay, imagine you're presenting it to someone who has never even heard of science before. Okay, how much of that thing do you need to explain? Number 6, state what the control is used for. So going back to the do not questions in lesson 9, your control variable is the one that stays the same throughout your experiment. And the reason for your control variable is to make sure that the experiment can be valid, right? So number 7 then asks, state how we know an experiment is reproducible. So reproducible means that you can do it again and again and again. So then what you need to do is you have to make sure that you can meet those same conditions so that you can recreate the experiment, right? So there's actually, so if we're going back to the water and the plant experiment that I've given you examples from, if someone were to do that experiment here in the UK, right, where it's the weather in the summer, where the weather can be hot, it's a little bit humid, right? Compare that to another person who does the exact same experiment in the Arctic, right, or the North Pole. Very, very different. Okay, so unless those conditions are the same, right, then that experiment will not be reproducible. Because you can already tell, right, your results of both experiments, one done in the UK and one done in the Arctic, are going to be very different, right? Because even though you give it the same amount of water, right, the conditions are still different, right? Even here in the UK, if you do it inside of a classroom compared to outside the classroom, there are so many different factors that can change your results. Okay, so you need to make sure that you add in every single detail to make sure that it's exactly the same as the original experiment. Number 8 then, state what control we used for the potato experiment. So you may not have gotten this one, but for the potato experiment, what we did is we used regular water instead of a sucrose or a sugar solution. Okay, and that's just to make sure that the experiment will still work the way it's supposed to. Okay, so for those 8 questions, please make sure that you have the correct answers, and we'll be moving along. And number 9, explain how villi, so remember that villi are the structures that are in your intestines, the one that looks like a mop. Explain how villi increase the diffusion rate of digestion in the small intestine. So that key word is going to be surface area. You need to have the word surface area in your response. The more villi there are, the more surface area there will be, which will increase the rate of diffusion. This means that more food is absorbed into your body. Okay? Very simple, very straightforward. Number 10, suggest why single cellular organisms, right, so bacteria, have no adaptations to surface area. So when you compare, again, yourself, a multicellular organism, to a single cell organism like bacteria, why do they not have the same adaptations we do? Right? And the reason is, well, 1, they can't grow into a bigger organism, but 2, they don't need to, right? Their entire organism, their entire body can have, is able to diffuse, okay? So they don't actually need a bigger surface area. They have the biggest surface area they can possibly get. And so in order to increase the rate of diffusion, they would have to become bigger, all right? And like so, they can't. Perfect. So just before I end the video, please make sure that you understand osmosis as much as you can. Watch the videos that are in the booklet, all right? And if those don't do any good, just search in YouTube, osmosis, all right? And you'll find a lot of really good videos on explaining what osmosis is, and diffusion as well, okay? But thank you for watching, and we'll see you next time.